privilege of having Robert Liu with us. Robert is training in physics and applied physics at Stanford all the way through, bachelor's, master's, and PhD, and then did his postdoctoral work at UCSF before he migrated from one coast to almost the other to join Emory, where he is on the faculty in the biology department. And going to be talking to us today about his work in the neuroscience of social interaction, social information processing, and learning. So, Robert, thanks very much for joining us.
All right, everybody can hear me okay? This is good? All right. So I want to start by thanking Bilal and Chris for the invitation to speak to you today. It's a real pleasure. I want to begin with a short video, and hopefully the audio will come off okay.
Johnny.
There we go.
Hey, Johnny. Look, it's Teddy. You want to play with Teddy? Teddy wants to play with you. Hi, Teddy. How are you doing? Yeah, Teddy. Johnny, look. Teddy, can you say hi to Teddy? Teddy needs a hug. Give Teddy a hug. Teddy, you want a hug? Teddy wants a hug. Teddy.
All right. You get the idea. If you were spending most of your time looking at this side of the video, you can pat yourself on the back and consider yourself one of the typically developing individuals in our population that prefer to look at this biological motion capture that is sort of regularly oriented and that sort of tracks the soundtrack that you're listening to, versus this side, which is the inverted and played in reversed version of that same video. So this suggests that we have this kind of preference for biological motion that facilitates our adaptive interactions with other individuals. But it doesn't have to be this way. It turns out that toddlers that go on to be diagnosed with autism actually don't have that kind of preference. Early on, they actually showed this kind of equal viewing time for both sides of these videos, suggesting then that there may be neurobiological mechanisms for processing social and non-social information that might not be identical in our brains. And yet, when we think about how the neuroscience of information processing has been studied over many decades, for example, to look at how what kinds of neural transformations occur from cues to behavior, what are mechanisms of learning and memory, as well as reward and reinforcement, almost all of these are studied in a non-social context, raising the possibility that we don't actually have a deeper understanding of what goes on when individuals actually interact and pay attention to social cues. So my lab has been really interested in the concept then of social information processing, trying to study these different areas, all in more social contexts. And the key then is to come up with experimental paradigms that use more natural social cues and behaviors so we can actually understand their underlying neurobiology. So I came up many years ago as a postdoc when I was at UCSF with what I called a kind of computational neuroethological approach to studying the neural mechanisms of social behavior. The general idea is something that we as neuroscientists often employ, the fact that some stimulus comes in and it drives some kind of response. In my lab, we tend to focus on these contexts in which this stimulus response is more natural and generally social in context. Then you want to study the sort of the neuro aspect of this computational neuroethology is to study kind of relevant neural circuits in the brain, apply computational methods to look at how the neural code may represent either stimuli or responses, and then use constraints from natural stimulus statistics or what we know about the behavioral responses and how they change to constrain our understanding of this underlying neural code. And in sort of modern day neuroscience now, we also can go a bit further and apply in rodents methods to test the causality of the kinds of mechanisms that we try to discover. So I'm going to tell you two stories today that use or that apply this kind of computational neuroethological approach. The first is an area that my lab has spent over a decade studying since I've been here at Emory and even during my postdoc. And I'll mostly review findings from my lab and others on the auditory processing of communication calls in mice. And the second story is a more recent collaboration with a Georgia Tech BME student in my lab who was jointly mentored by Larry Young, studying social bonding in monogamous prairie voles. But first, the auditory processing of communication sounds in mice. The big message that I want to convey is that learning a sound's meaning alters the feed-forward coding of those sounds to improve their functional processing. Now, this is work that actually spans many years, beginning with a postdoc, Edgar Galindo-Leon, and several graduate students, two of which are Georgia Tech BME students, Frank Lim, who's a Bio-E student, and now, most recently, Kelly Chung, who's in the lab, who's a BME student. All right, so to introduce this topic, I played back a Chinese poem, which, if you don't know Chinese, may have just sounded like a bunch of words that were exactly the same. But if you do know Chinese, you may have recognized those as the words of part of this poem that says, "Lion-eating poet in the stone den." It's different intonations of the same basic sound to some degree, s. And the point is that people who recognize or understand that language actually very immediately understood how to parse those sounds and discriminate one from the other to create an understanding of the words. So what is changing in the auditory system to support that immediate recognition of those communication sounds? That's what my lab has been interested in. And actually, more generally, many labs are interested in this concept of neuroplasticity. What changes when your brain is able to acquire a new skill, recognize a new stimulus, etc.? And the basic idea here is that when a sound category is initially novel, unfamiliar, and occurring in some kind of behavioral context, in the case of the auditory stimuli, it goes on and elicits responses throughout the auditory neuroaxis, all the way up to auditory cortex, while this contextual cues may stimulate your other modalities. And then together, they combine to feed into sort of perception and action areas that drive your behavioral response to that sound in context. Now, that behavior may have a consequence, whether it's appetitive, aversive, or potentially just purely neutral. No matter what, this will occur with some neuromodulation that bathes your brain, bathes your neural circuits, whether they're cholinergic, noradrenergic, or maybe even neuropeptide hormones like oxytocin or estrogen that are co-occurring. And that neuromodulation has effects widely throughout the brain, but also within the auditory system itself. The idea then is that this is something many people are actually interested in, that sort of dynamic modulation of neural activity by neuromodulators. But I want to point out that these also have lasting effects, because all of these neuromodulators also affect neuroplasticity. So that when that sound category is encountered again in the future, when it's more familiar, and even when it's out of context, its ability to drive perception and action is now improved. So that's the underlying hypothesis, that learning induces some long-term changes that bias a sound's feed-forward processing. But exactly what are those long-term correlates for sound memories that are acquired through our experiences? Now, a few years ago, the sort of classic lore was that use it or lose it. You have map expansion. You create more territory in your brain to represent relevant stimuli. And this is just an example from classic work by Greg Reckenzone in Mike Merznick's lab, where a monkey that is initially naive to a task has a representation of a frequency around 2.5 kilohertz that's relatively small. But then you train that animal to discriminate sounds around 2.5 kilohertz, and its representation of that frequency increases or is enhanced. So for many years, that kind of map expansion, the map, when I say map, I refer to the tonotopic map of sound frequencies within the auditory cortex. That expansion was thought to be the sort of correlate of learning about a sound. A few years ago, though, Michael Kilgar's lab, who actually played a big role in our understanding of this kind of receptive field plasticity, found that long-term cortical map plasticity can actually decay without affecting behavior. So actually, map expansion is not a long-term correlate. It may actually be useful for learning, but is not itself its memory trace, if you will. So then what are the long-term correlates of learning to functionally detect, discriminate, categorize, or recognize, in our case, more natural social vocalizations? Well, for that, we have turned to a mouse maternal model that we hope to champion ever since I was a graduate student, or a postdoc rather, in Michael Merznick and Christoph Schreiner's lab at UCSF. In this model, we have mice that actually naturally emit a communication sound that's in the ultrasonic vocalization range. In particular, mouse pups emit these USVs that elicit what's called a maternal retrieval, which you'll see here in this short video. You'll be able to hear ultrasonic vocalizations through a bat detector in the corner here. Okay, so those are the calls. You saw the mom response, go and pick up the pup and bring it back to the nest. It's a very robust behavior. And if you look at the actual vocalizations that elicit this response, they actually form acoustic clusters in certain acoustic parameter spaces, like duration and frequency. The calls themselves are very simple single frequency whistles, like what you show here. And they're somewhat similar to actually calls made by adult males in the presence of females. But there are also differences. As you can see here, these calls tend to be more frequency modulated. And if you look at a large population of calls, they actually form a distinct cluster in this acoustic space from what we see for pup vocalizations. Now, this kind of acoustic categorization is reminiscent of what we see for human speech. For example, in human speech, vowels, if you characterize sounds according to their formant frequencies, they also form these kinds of acoustic clusters. So then this gives us an opportunity to look at how neurons within the auditory system respond to specific acoustic features that define sound categories. In this context, the mouse model has further advantages, which is that there seems to be a learning component to this recognition by maternal females. First, if you just take mothers and play back pup ultrasonic vocalizations or neutral sounds, mothers prefer to approach USVs about twice as often as compared to the neutral sound. But naive virgins don't. They go to both sides about equally often. On the other hand, if you give those virgins 5 days of experience caring for pups with the litter, with the mother, it too begins to show this preferential approach to pups, suggesting that this preference can be acquired through experience. So that opens this great opportunity then to use this very natural behavior to understand what changes within the sensory system in order to support this recognition. So now I'm going to review a bunch of findings from my lab and others that have led to some insights about this sort of underlying memory traces. But first, in a collaboration with actually one of Garrett's former students, Nick Lessica, we actually found that even before you look at mothers, if you just look at naive animals, there is already an intrinsic bias for ultrasonic sounds in mice. In our case, we use the CBA mice. And that's reflected here. We saw this, what we call a kind of central magnification of ultrasonic frequencies within the midbrain of the mouse. And that's shown here where higher frequency sounds elicit a much greater response in the midbrain compared to the auditory nerve, whereas lower frequencies did not. Now, Nick's lab further took microelectrode shanks and recorded systematically in the auditory nerve, cochlear nucleus, and then in inferior colliculus, and replicated these results using unit recordings, suggesting that perhaps there's a kind of acoustic fovea that emerges in the midbrain for these high frequencies. By the time you get to the auditory cortex in the mouse, there's actually its own field, an auditory field that's dedicated to ultrasonic processing, suggesting a further sort of elaboration of this frequency range. Now, some of the earliest studies that we then went on to show, first at UCSF and then continuing in my lab here at Emory, was that as you compare naive animals to maternal animals, there are long-term forms of plasticity for those natural pup vocalizations. For example, if you look at the ability of neurons to follow the natural 5 Hertz rate at which mouse pups produce pup calls, in mothers, they're better able to do that compared to naive animals. If you look at the ability to convey information about individual calls for, say, detecting or discriminating them, mothers show enhanced information compared to naive animals. But if you look at that classic phenomenon, map plasticity, and you ask whether or not mothers show an enlarged representation for calls compared to naive animals, we actually did not see that occur. So there was no tonotopic map plasticity for ultrasounds, whether we looked at frequencies, vocalizations themselves, or vocalizations themselves. Instead, what we observed in both anesthetized as well as awake recordings is what we call an enhanced lateral band suppression, where neurons, when you play back pup ultrasonic vocalizations, say during this green period here, instead of being excited by the calls, are actually being suppressed by the calls. And that suppression, interestingly, was stronger in maternal animals than in non-maternal animals, but specifically in fields that were tuned to frequencies lower than the natural ultrasonic frequencies, almost suggesting that as the animals are listening to these calls that are all in the 60-some kilohertz range, those areas that are tuned to lower frequencies are actually being more strongly suppressed. And that was more recently confirmed in a completely different set of experiments using different animals, different technique, where we tracked how neurons responded to playback of pup calls from naive animals through motherhood to post-weaning, and saw a progressive decrease in the firing rate, specifically in these lateral band areas, suggesting that this kind of contrast enhancement may be a real effect for improving the ability to recognize these calls or maybe detecting them in background noise. So the picture that emerges looks something like this. As you go from a naive animal listening to pup calls through motherhood to post-motherhood, rather than this kind of picture where you have an enlarged representation of high frequencies shown here in red, you actually have this kind of contrast enhancement where lateral band areas are more toned down in their response. But I don't want to give you the sense that all of the changes are all about inhibition, right? There are changes in how neurons are excited by calls as well. And so next I'll review some findings about how neurons are excited by ultrasonic vocalizations. And first of all, I'll point out that even within the brainstem, we actually found subcortical plasticity as you go from a naive animal to a mother in responses to ultrasonic frequencies. When you get up to the cortex, we used a modeling approach to discover that neurons within auditory cortex can actually respond quite faithfully to the onsets of these ultrasonic vocalizations if we model their responses based on how they respond to pure tones. And for that kind of model, we used basically a peripheral model of sound transduction and kind of conveyed it up to the cortex. And what was interesting in that was that those neurons that were best predicted by our model turned out to be almost entirely putative interneurons, meaning that they are putative GABAergic interneurons, suggesting that the first response in cortex that's very reliably acoustically driven is actually an inhibitory response. Those neurons that were more likely to be putative excitatory pyramidal neurons actually tended to be very poorly predicted by our model, suggesting they weren't actually following the acoustics at the onset of the sounds. So what's going on? That's when we looked more closely at what changes during motherhood. So then if you compare non-maternal animals to maternal animals and how they respond to these pup calls or to acoustically matched adult calls that are matched in their onset properties, non-maternal animals actually respond about the same to both pup and adult calls. But maternal animals actually preferentially respond more strongly to pup calls over their acoustically matched adult calls, fitting with this picture of this enhanced bias for pup vocalizations when they've become behaviorally relevant to the mother. How exactly are these neurons able to do that? Remember, these calls are all matched in their onset properties, and that led us to think about maybe these neurons are actually responding to the physical trajectory of these vocalizations across time. How does the frequency change across time? And in fact, we've recently found that the offset responses in neurons actually encode this kind of pitch trajectory and change. So to show that, let me point out first that in cortex, in auditory cortex, like many other parts of sensory cortex, there's a huge heterogeneity in terms of how neurons respond to stimuli. So for example, here's a neuron that as we play back short ultrasonic vocalizations, you get responses that are, in this case, quite strong right at the onset of a sound, but linger even after the sound comes off. And you see that more clearly here as we lengthen the USVs. In all of these cases, there is a portion of the response that is there during this on portion of the sound, as well as a portion of the response that appears after the offset of the sound. So we call these types of units on and offset responding. There are units like this that only respond during the on portion of the sound, as well as units like this that respond just at the offset, and only at the offset of very specific calls. All right? The different rows here are different calls with different acoustic properties. Now, what was really cool is that if you look at the prevalence of these kinds of offset responding neurons, that actually changes with motherhood, with an increase in that prevalence for offset only in maternal animals compared to non-maternal animals. And that's almost entirely due to this thick spiking population of putative pyramidal neurons, rather than in this thin spiking population of inhibitory neurons, further suggesting that it's these pyramidal neurons that generally provide the output of cortical processing that are retuning to follow this sort of acoustic trajectory. Okay, so that leads to this kind of model where in addition to this kind of contrast enhancement, we have individual neurons actually scattered throughout auditory cortex that are now responding better in mothers to pup calls over adult calls. So now, how does this kind of transition occur? What actually happens during motherhood to allow for this kind of plasticity to arise? And to understand that, I'm going to briefly review some more recent work that's both preliminary in my lab and that was also recently published from other labs, looking at biological mechanisms for learning in this context. So my lab has long hypothesized that estrogen priming of sensory plasticity within cortex may be a mechanism for enhancing this. And I won't show this today because of time, but we now have preliminary data suggesting that estrogen, along with social interaction with pups, increases BDNF expression within auditory cortex. For those of you who know about BDNF, it's this neurotropic factor that's strongly implicated in sensory plasticity in sensory systems. Other labs, so for example, Adi Mizrahi's lab, has shown another kind of mechanism that allows for this plasticity to emerge. And he showed, for example, that pup odors, a kind of multimodal interaction between pup odors and auditory processing, can be found specifically in the auditory cortex of mothers, but not virgins. So when you present the odors of pups along with pup calls, the odor actually can modulate firing within auditory cortex. But again, this doesn't happen in naive animals. They've gone on, his lab has gone on to show that there's also a kind of disinhibitory mechanism during learning, where pyramidal neurons, when you present the odor, actually get more excited in response to these vocalizations, while interneurons are actually being suppressed in the presence of those odors. So that suppression of inhibition is thought then to allow excitatory responses to increase, potentially to allow for plasticity to occur. And what's really cool is that Rob Fromke's lab a couple years ago demonstrated a possible mechanism, a neuropeptide oxytocin, that may actually mediate this. In particular, pup odors may be releasing oxytocin within the brain, this sort of social neuromodulator, and operating directly in the auditory cortex can lead to a kind of disinhibition, like what Adi Mizrahi's lab showed, where a presentation of oxytocin weakens inhibitory synaptic inputs while eventually enhancing excitatory synaptic inputs. Over time, then, this could cement a longer-term change in how these neurons respond to these ultrasonic calls. So this is our kind of working model, then, for how the auditory cortex learns about the importance of new sounds and reflects those changes in how these neurons respond. So we have this kind of model where a lot of neurons, pyramidal neurons, receive inputs from many other excitatory neurons, but also from inhibitory neurons. Pup odors, perhaps acting through oxytocin release, then leads to a disinhibition of pyramidal neurons by inhibiting putative interneurons. So that disinhibitory effect actually is something that other labs have found in other types of learning contexts, such as in a fear learning context. In addition to this kind of disinhibitory mechanism for learning, our results also suggest that there's this kind of neural population contrast that is arising from a strengthened feed-forward inhibition in specifically lateral band areas. All right. So that's how we're thinking about this model now, and there's lots still to do. But if I could give a kind of summary of some important points I want to come back to later on. One is that there's this kind of innate sensory circuit predisposition, this acoustic phobia that we think we've discovered for processing evolved communication signals. But then social experience also enhances the sensory coding of those vocalizations through neuroplasticity mechanisms. So this idea of predisposition and plasticity is something I want to come back to in the second system that I want to talk to you about, which is this model of social bonding in monogamous prairie voles, focusing particularly on this idea of social reward and reinforcement. And this is work that essentially shows that dynamic corticostriatal activity can bias the formation of a social bond in these prairie voles. And this is work that was pioneered by another Georgia Tech BME student, Liz Ann Amadei, in collaboration with Larry Young. And this is being followed up by Jim Kwan, who's an Emory neuroscience graduate student. So pair bonding, we're making a big transition here, so I wanted to give you a little bit of an idea of why this is an important topic. And for those of our generation, this is probably a familiar movie to you, Titanic, very strong pair bond, right? Well, that kind of pair bond happens in about 5% of mammals. It's a reproductive strategy that's quite successful in about 5% of mammals. But exactly what happens in the brain that allows you to go from this kind of non-bonded state to a bonded state? One idea that's been out there is that there's kind of over time an increasing reward value that's placed on your partner. So that leads us to ask, well, how really is the mesocortical limbic reward system activated and modulated during the formation of a bond? And to do that, we're going to use this prairie vole model of social bonding, which has been pioneered by Larry Young's lab, initially working with Tom Insel, and now many other labs have sort of embraced this as kind of the canonical model of social bonding. The general idea is that when you put a male and a female prairie vole together and they cohabitate, after enough time, they form a kind of social bond, which can be tested in the laboratory using what we call a partner preference test, where you give the subject the chance to spend time huddling, sort of motionlessly, with its partner, aka cuddling, if you will, or spending time huddling with a stranger. And you find that in prairie voles, unlike other types of closely related voles, prefer to spend more time huddling with their partner than their stranger. And so this is the laboratory signature of a pair bond formation. Interestingly, when Tom Insel and others went to look at the brains of prairie voles and compared them to non-monogamous voles, they found that oxytocin receptor expression was very different throughout key parts of the brain, in particular prefrontal cortex and nucleus accumbens. And when Larry went in and injected an oxytocin antagonist, site specifically into either the prelimbic part of prefrontal cortex or into the nucleus accumbens, he could reverse or abolish this kind of partner preference that was seen in control animals, suggesting that oxytocin acting specifically in those brain areas is important for bond formation. Now these two areas are part of a larger circuit involving dopamine and oxytocin projections that we think is critically involved in social bonding. But just knowing that those areas are involved, where those sites are, doesn't really tell us how those areas communicate with each other during social interactions leading to a bond formation. So what exactly are those sites' modes of action? To study that, we started to do neural recordings in prairie voles by implanting what we call a neural logger onto the brains of skulls of female prairie voles as they cohabitate with a male prairie vole. We videotape them as we're recording so that we can synchronize neural and behavioral activity. And then we have lots of undergraduates as well as graduate students watch those videos, kind of a vole porn, if you will, and score them for different kinds of behavior, like mating, self-grooming, and of course huddling, which is the expression of the bond. As those behaviors occur throughout this 6-hour cohabitation time. So here's like an epoch where the male is mounting the female and mating. Here's an epoch where the female is just self-grooming by itself. And then here's an epoch where they are just sitting side by side and huddling. So once we had this behavioral records, we could look at, well, when do these behaviors begin? How long do they last, et cetera? And one of the things that emerged quite quickly is that mating happens very rapidly when you put those animals together, but huddling tends to happen much later. Not only that, huddling is quite different from one individual to another. And that's shown here in these huddling trajectories for individual voles, where each line reflects when an animal begins to huddle and how long it's huddling. So you see, there's quite a lot of variability. If we mark a particular time point to accumulate, say, 5 minutes of huddling time, that gives us a kind of measure of the latency to begin huddling. And if we look at that huddling latency and compare it to how much time animals actually spend huddling by the end of the 6-hour cohabitation, they are very strongly correlated, which kind of makes sense, right? The earlier animals begin to huddle, the longer they're going to huddle. But it turns out when you look at other behaviors, that relationship between timing and duration doesn't necessarily hold. So this was an interesting and important finding, and suggests that we could use huddling latency as a kind of measure for how quickly a bond gets formed. So what gives rise to that huddling latency? Well, we looked at possible correlations with a number of different behavioral measures, including latencies and durations, et cetera, and none of these could explain this kind of huddling latency. So that led us next to take advantage of our neural recordings and look at potential relationships between this huddling latency and measures of neural activity within the 2 areas we placed electrodes, medial prefrontal cortex and the nucleus accumbens. And those recordings, an example of those recordings are shown here. We recorded local field potentials and divided them into various epochs that were synchronized with the behavior. So epochs of mating, self-grooming, and huddling, for example. And if you just look at what the power spectrum of those local field potentials look like within the MPFC or nucleus accumbens, we see that there are oscillations in that local field potential. And moreover, those oscillations can be modulated by behaviors. In particular, what Liz Ann sort of locked on and noticed was that this very high low frequency peak at about 5 hertz, which is kind of roughly a theta rhythm, there was a very strong modulation of this peak across different behaviors. And so that led us to look more closely at different measures of how these brain areas might be talking to each other. That peak, you can see, is apparent in both MPFC and in nucleus accumbens. So we wanted to know whether those might be actually related, and whether activity in one brain area could actually affect the activity in the other. And so we used a measure of what we call functional connectivity, called the cross-frequency coupling. The idea is that we take the signal from one brain area and filter it to represent, say, the low-frequency component, and then take the other signal and filter it to represent the high-frequency component. And if you can squint and see sort of that there's a phase relationship between when, what phase the MPFC low-frequency signal is at and the amplitude of the gamma oscillations in the nucleus accumbens, that relationship turned out to be actually quite consistent. There's what we call a cross-frequency coupling, where MPFC low frequencies around 5 hertz modulates gamma activity in the nucleus accumbens at about 80 hertz. And so this is an effective channel of communication between those two areas. So now with that channel, we can just assay that cross-frequency coupling across the whole cohabitation, across all different kinds of behaviors. And that's shown here in the color-coded fashion. And you can kind of squint and make out that, yeah, there's kind of changes across time. That mating tends to give very high cross-frequency coupling, but huddling tends to actually give you kind of low cross-frequency coupling. Now, this is one animal's net modulation across time. This is another animal's. And you see, though, that there's a lot of variability across animals. And in fact, if you look across all of these animals and look at their cross-frequency coupling during different epochs of time, whether animals are huddling, whether animals are in this baseline period where they're just sitting by themselves, or whether they're in this non-huddling behavior, which essentially we classified as a more active behavior whenever animals are just not sitting next to each other. And you see that there's a lot of variability, but first of all, huddling, which is supposed to be this rewarding thing, actually does not elicit activity within this reward pathway, which is a little bit of a surprise to us. But instead, during these non-huddling activities, there's a lot of variability across individual. And moreover, it correlates really strongly with this huddling latency, suggesting that this brain activity, this functional connectivity between these two areas, was telling us something about how quickly animals will go on to express their affiliation. Now, this was a correlation that we observed when we took neural data across the entire cohabitation and compared it to the latency to huddle. But it actually emerges across time. And so when we started taking data, looking at the data in shorter epochs from the onset of cohabitation, we found that even at about an hour into cohabitation, there's already nearly a 0.75 correlation. But even before that, even at time 0, which reflects a time point where the animals have not even met, there's already a high correlation, suggesting a kind of predisposition based on the neural activity in this pathway for animals to be kind of sociable with another individual. But then that actually increases over time. And if we look in a non-hit circuit, there's actually no such correlation. So this is specific to this medial prefrontal cortex to nucleus accumbens circuit. And this correlation seems to increase over this first hour, which is the period during which mating first begins. So that led us to look more closely at what happens to this functional connectivity around mating. And so this is mating right here. Before mating, this is one animal where there's not much net modulation. And after mating, there's a slight bump in this animal for a few minutes afterwards. Here's another animal where you get a much bigger bump in their cross-frequency coupling. Here's another animal where you get almost no bump. And when you look at all the animals together, you kind of see that there's a color gradient that can be observed here. And the coloring is actually based on how quickly animals huddle after they come out of mating. And in fact, if you look at the change in that net modulation and correlate it with that huddling latency from coming out of mating, there's actually a pretty strong correlation, suggesting that how these brain areas are communicating as they come out of their first mating event is saying something about how quickly they want to come together and express their affiliative behavior. And that is significant for about a couple of minutes after they come out of mating. And even if you subtract out that baseline correlation I mentioned earlier, there is still a significant correlation between these two, suggesting this is a separate effect than any baseline predisposition to be sociable with each other. On the other hand, if you look at our control behavior, self-grooming, there was no such enhancement that was correlated with their huddling latency. Okay, so that suggests mating is this experience that can change how the brain responds to the partner, which then correlates with how quickly they're going to be affiliative. Can we now actually see if it's causally involved? So for this, we used the channel rhodopsin and optogenetics approach to try to bias the formation of a bond in prairie voles that would not normally form a bond. And so this just shows the expression of channel rhodopsin within the nucleus accumbens. We injected our virus into the medial prefrontal cortex and put a fiber over the nucleus accumbens to excite the projections into nucleus accumbens. And then we allowed the animals to cohabitate only for a limited time, 3 hours, in a way in which normally bonds would not be formed. The male was actually jailed, sitting under a cup, unable to physically mate with the female. But every time the female walked into the social chamber, it got a 5 Hertz stimulation, optogenetic stimulation of these inputs from MPFC to nucleus accumbens. And then, so if we look at how these animals are stimulated compared to control animals that just got a fluorophore virus that does not actually gate activity within the transfected neurons, there's no difference in terms of how much optical stimulation they got or in terms of how much time they spent in the different compartments of this cohabitation chamber. But then, when we took them to the partner preference test, when we looked at our control animals, most animals actually showed a preference for the stranger over the partner, consistent with the idea that our cohabitation paradigm does not lead to a bond formation. On the other hand, those animals that received channel rhodopsin showed a preference for the partner over the stranger. And when we looked at the difference in time for huddling with the partner versus the stranger, there was a significant effect with a preference in the stimulated animals for spending more time with the partner. So this theta frequency oscillatory drive can, into nucleus accumbens, can causally bias the emergence of this affiliative behavior. So this puts together our kind of working model then of what's going on. The medial prefrontal cortex at low frequency phase modulates the nucleus accumbens in the gamma frequency range. And that strength of modulation actually varies across subjects and correlates with the huddling latency. But first mating actually can improve that brain affiliative behavior correlation. And showing causality, directly enhancing that activity can actually shift the preference towards a partner. All right, so now stepping back and trying to put this together with the auditory story I told you about, the big picture insights that we're gaining from this model are that, again, we see this kind of individual predisposition, this time in the cortical limbic circuit, for sociability, but that social interactions can dynamically enhance the ability to switch how an individual responds behaviorally to another individual who is presumably a source of social reward for that individual. And that brings me to a recent paper, specifically this idea that keeps reoccurring of predisposition. This was published by Warren Jones and Ami Klin from the Marcus Autism Center down the road from Emory earlier this year, which found that there are individual predispositions for seeking out social information. And they did this by looking at human infants and looking at how they view videos of social interactions with caregivers, with other infants. And amazingly, when they compared monozygotic twins to dizygotic twins or other age-matched kids, in monozygotic twins, there is a strong correlation between one twin and the other in terms of, at a macroscopic level, how it is spending its time viewing those videos, specifically how it's spending time viewing social information, like time spent looking at the eyes and also the mouth. There's a strong correlation there from one twin to another that's present in monozygotic twins but not in dizygotic twins or others. This is at the macroscopic level. More amazingly, when they looked at the individual traces of saccades, visual saccades, how they actually viewed videos, monozygotic twins demonstrated greater probability relative to dizygotic twins of moving their eyes at the same times. For each saccadic eye movement by twin 1 within 350 milliseconds, there was an 18.6% increase in twin 2's probability of also making an eye movement, suggesting that there's an innate, genetically potentially driven salience for social information that's there from birth and actually is preserved up to, I think they looked at 31 months. There is still a strong genetic drive for how they view these videos. So that predisposition thing, something we often ignore in neuroscience, actually seems like it could be a key for understanding individual differences and eventually making the jump to potentially mental health. All right, so then stepping back, I hope I've been able to demonstrate how this kind of computational neuroethological approach to social information processing has given us some new insights, new lessons. First, in this case of auditory processing and communication calls, it suggests that actually, instead of map expansion, we should be thinking about different ways of enhancing at a population level the neural contrast for how sounds that are communication sounds or behaviorally relevant are represented in the brain. And in the social bonding case, that we should focus on this corticostriatal circuit that may be helping to switch animals towards more affiliative behavior, which kind of fits in the language of how people study this corticostriatal circuit, which is often in the context of drug addiction and also habit formation. Maybe love is like forming a habit for your partner, for example. All right, and with that, I'll end with acknowledgments, especially funding from NIDCD for our auditory work and NIMH for our social bonding work, collaborations with Larry Young, and also Edgar, Frank, Katie, and Kelly, who were so important for a lot of the electrophysiology in the auditory case, and Liz Ann Amadei here, and Jim Kwan, who were involved in the social bonding studies. And with that, I will end.
People may have to run off to the next class, but we have time maybe for a brief question.
So you showed that this theta modulation of medial PFC to nucleus accumbens changes the preference, partner preference. Do other frequencies help? Does it matter?
Well, I don't know the full answer yet, but I can give you a couple of anecdotes. One is that in a few animals, Liz Ann actually tried stimulation at 20 hertz. Those animals actually developed a strong stranger preference. So it may actually be that frequency does matter. Yeah. But we haven't followed up on that. That was only a couple of animals. Hey,
That's very interesting. I enjoyed your talk. So we do a novel object recognition task, and there's a version of it which we haven't done, which is pups with mothers, and the mothers they recognize the pups, and then they give them pups to other pups. I always thought it was very odor related. Is there a chance that those pups have different vocalizations?
So first of all, in rodents, odor and maternal behavior are is critical, very important for maternal behavior. In our model, how I think of it is odor is kind of the innate response, innate signal that these are pups. If you're a mom, be maternal. But the auditory system and these vocalizations are additional cues that help a mother locate pups and respond to them. And so I think of the auditory system as kind of learning from the olfactory system about the cues of the pups. And that's where the odor modulation of auditory cortical activity that may be mediated through oxytocin is a really exciting possibility. As far as individual vocalizations from different pups, in mice, there's no clear evidence that individual pups matter. Sex of pups may matter. So in rats, males versus females, mothers seem to like to pay more attention to the males than the females. There were some studies done by Peg McCarthy's lab in University of Maryland that suggest that male pup vocalizations are particularly interesting to the mothers. But in terms of individual animals, when pups are really young, there's a lot of individual variability in their vocalizations, but as they mature, after about P13, which is when their ears actually open, P11, P13, their vocalizations become more similar to each other in certain characteristics. And just, ecologically, it may be not, these mothers have a lot of pups all at once. They will actually co-care for pups of their sisters pretty easily. They'll harem raise pups. So it may not be so important for them to invest time into an individual pup the way it would be for humans, for example.
I was wondering, you talked about maternal mice versus
Non-maternal.
non-virgins. What about the adult males? How do they react to pups?
So adult male mice can eventually show a preference for pup calls, but they take longer. So they have to have more than one litter of experience before they'll show that recognition. They can show it, but it's just longer. So, yeah. And that makes it logistically harder to do experiments on them. So we haven't really investigated their neural responses as much.
All right, maybe we can take the rest of the questions offline. Thank you again, Robert.